Hello dear students, welcome to the another video tutorial of parasitology. And in this session we are going to get information about the life cycle of the Ascaris lumbricoides. Okay, so this will be the part third of our Ascaris session. Okay, so let's continue with the life cycle. Before this lecture, we have taken idea about the uh, reproductive organs and the how that particular parasite is reproduced, okay, and that reproducing genitalia of um, helping in the reproduction. That process, the two genitalia of uh, as well as male and female, that is the two distinct sexual dimorphism we have seen. Okay, so let's start with the life cycle. The very first process is copulation and fertilization. How that eggs get fertilized and uh, acting of a copulation uh, process. Okay, so the ascaris is uh, copulates in the intestine of a host itself. Okay, and the sperms by the amoeboid movement reach the seminal receptacles of the uterus and fertilize the ovum. Okay, so after the fertilization, these fertilized eggs are passed one by one into the uterus. Okay, and that uterus is going to store these fertilized eggs, as we have seen. A sphincter or the sphincter muscles, okay, the sphincter muscles are regulates their passage from the seminal receptacles into the remaining part of the uterus. Okay, so these mammaliated eggs, that means it having the fertilized eggs, that are known as the mammaliated eggs. So immediately after fertilization, the cell membrane of a zygote separates from its cytoplasm and thus becomes a fertilization membrane. Okay, so soon it is fortified, okay, or the transferred by protein, or the coating by a protein and glycogen granules is granulated and by the cytoplasm. So thus it transforms into a thick and hard chitin shell. Okay, high chitin means it is a calcareous cell, and this something is chitin is a substance something like the calcium. Okay, and this made up of the pure calcium. So this chitin shell is thickened around, and that protein is covered on this particular fertilized egg. Okay, and that is the covering which is not get digested into the uh, our stomach acids and the digestive juices into the stomach itself. So it can't be digested or can't be dissolved by means of this acidic medium into the stomach. That means it is the um, modifications in this particular um ascaris parasite. Okay, so it have evolved that particular characteristics to get survive into the this tough type of the situations into the host stomach. Okay, as soon as it passes with the uh, stomach to the duodenum or the small intestine, where there is an alkaline medium, so that alkaline medium is dissolving that particular egg shell or the chitinous shell, and that shell, after that the um, fertilized uh, ovum is going to release into the lumen. Okay, so this is very interesting part, that it is evolved and it can't be get dissolved by means of the acids of our stomach, like HCl and something proteolytic enzymes. Okay, so soon it furthered, and the thick hard chitin shell is formed. Okay, so it is the one type of the protections which have evolved by means of this particular type of the Ascaris lumbricoides. Okay, so the cleavage and early embryonic development into the intestine. Okay, that embryonic development is possible in Ascaris only outside the body of a human host in the soil. Okay, as we have seen, the fertilized eggs are stored into the uterus. Okay, after that it comes to the uteri, and that uteri is going to uh, pass these eggs into the soil, and in the soil this particular process known as the cleavage and early embryonic development happens into the soil. Okay, so the in the Ascaris, the embryonic development, or the early embryonic development, is occurred into soil, where there will be required a low temperature, more oxygen, and suitable moisture to get um, convert into fertilized egg to the embryo. Okay, and that embryo is ingested by means of the host. Okay, that will be the small childrens, or the human host, or the other um, mammals. Okay, and after the contaminated soil, or the fruits, or the our vegetables get contact with the, or the raw, if we eat them raw. Okay, we do not maintain our hygiene standards, then these embryos of these particular um, are get ingested by means of uh, the that host, and there will be the cleavage is, and that cleavage of this particular embryonic development will be holoblastic cleavage. Okay, and there are the particular spiral and the determinate type of these holoblastic cleavage. Okay, so in quite early stages, these different blastomeres of the embryo becomes emarked. Okay, and to form different organs of the future juvenile ascaris. Okay, and these future juvenile ascaris will be the stage where it can infect the host. Okay, so it will be covered under the topic infection of host. Okay, so these ascaris has a single host life cycle, that is monogenetic life cycle. Okay, and as we have seen in the entamoeba, that are the digenetic life cycle. Okay, so as in the Ascaris, it is the monogenetic life cycle. So egg contain the second stage juvenile are called embryonated eggs, and these embryonated eggs are infective to human host. Okay, so the people acquire infections by ingesting embryonated eggs with the contaminated food or contaminated drinking water. So when these eggs reach into the intestine of a human host, there is coverings are digested, and their coverings are made up of the shells, calcareous cells. Okay, and that are get digested, and the juveniles is measuring become free in the intestinal lumen. Okay, not in the stomach, free in the intestinal lumen. So boring through the intestinal wall, okay, and that these juveniles make their way to the submucosal blood capillaries, okay. Then these juveniles are entered into the bloodstream, okay, where these circulations and these blood circulation, these undergo an extensive migration in the host body. So they are migrating into the host body, okay. So after complete migration into the various organs, so that will be divided. This migration is divisible into two phases. That are, the first one will be the primary migration. In that, during this migration, the juvenile remains into the blood of this host for about a week. Okay. After a week, where there from the capillaries of intestinal wall, and these reach into liver with the blood flow, okay, and in liver it stay about the 3 to 4 days. Via the hepatic portal system it enters into the liver, okay, and then it another 3 4 days these reach to the lungs via hepatic veins, okay, and it may reach to the heart, pulmonary arteries, okay, where the secondary migration of these particular juveniles are happens, okay, and these migration take place in the respiratory organs and the alimentary canal of the host, okay. But sometimes what happens, these migrations are not well proceed, and it may accidentally migrate to the brains, and this will be categorized under the third category we are going to see forward, okay. Let's focus on the secondary migration again, okay. So the in lungs, these juvenile make their way out from the blood capillaries into the alveoli. Alveoli, the air sac of the lungs, where the oxygen exchange is happen, or the gas exchange happens, okay. So these alveoli, the fourth stage juvenile stars ascending up to the bronchi, okay, bronchi, where the two separates air is separated to the two air sac or the lungs, okay. At that bronchi it get migrated to the bronchi after the alveoli, okay. And this fourth stage of juvenile start with the and get migrated to bronchi, okay, and it trachea, okay, of the host, okay. So therefore that host is suffering from the coughing, okay, for these reasons, and hence the juveniles are cuffed into the pharynx, okay. With that cuff these juveniles are get carried, okay, and into the pharynx of the host, and they are swallowed into the esophagus, okay. If that host is going to swallow that cuff, it going to entered into the esophagus and that carried into the intestine, okay. The juveniles soon undergo the fourth and the last molt of the cuticle and become the adult. This means the circulation is completed over here when that juveniles are going to um, pass through the liver, lungs, okay, then with the bronchi, and again come with these cuff to the our esophagus or the mouth, and where that get swallowed and again come into the stomach or the intestine. So one circulation is completed within the 8 to 10 weeks, okay. So after reaching back into the intestine, the juveniles soon undergo the fourth and the last molt of their cuticle and their cuticle and become adult, okay, where there formation of the cuticle on that particular juveniles, okay, and where within the 8 to 10 weeks they are becoming adult, where these ascaris starts reproducing into the intestine. So its lifespan into the host is about 9 to the 12 months after the infection, okay. After these it dies and get disintegrates, okay. So I have told you that if this migration is not done with the proper way, that may leads to the aberrant migrations, okay. So sometimes the juveniles migrate into the brain, spinal cord, eyes, or etc., which is not the path of these particular juveniles to get migrate proper path, okay. So with the blood of the heart instead of migrating into the lungs, okay. Sorry. So in such organs these soon become dead and insisted in the calcified cyst, okay, where these juveniles get dead and the calcified cysts are uh, deposited over this particular aberrant migration process into the brain, spinal cord, or the eyes, such other organs than the which organs are comes into the path rather than these organs, okay. So similarly, during their migration from the trachea into the pharynx, okay, the strong coughing may cause uh, injection of the juvenile through the nose or the mouth of the host, okay. And Ascaris infections can be prevented by maintaining the cleanliness and good sanitary conditions, okay. Our vegetables should be thoroughly washed or preferably in mild solutions of KMNO4, okay, and properly cooked before use. Washing the hand with the antiseptic soap before the touching edible materials may help to reduce the infections of the ascaris. So as you can see, the infections like the entamoeba or the other stomach infections or the stomach problems are reduced in these pandemic days, okay. Why these are reduced? So everyone is going to wash their hands and maintaining the cleanliness of a hand, or the sanitization process is reduced. Ultimately the patient numbers which are suffering from the stomach problems or the entamoeba histolytica or the dysentery problems, okay. So these number are reduced by means of the taking care. Likewise, this care is should be taken in this particular type of the uh, infections or with the ascaris or the parasitic infections, okay. So this will be the again explanation of the life cycle, okay. As you can see, the adult in a small intestine as of highlighted with the first, and this image is taken from the CDC, that is the Center for the Disease Control for the USA, okay. So as listed in the second is the, as you can see, the numbers are highlighted the processes, okay, the steps of this particular life cycle, okay. As you can see in the second step, that will be the unfertilized egg will not undergo the further development, okay. Unfertilized means um, that unfertilized egg is will not go and it is degenerated and it will remain in the dormant conditions in the soil, okay. After the with fertilized egg is passed into soil by means of the fecuses, that fertilized eggs gives into the undergo into the further development, okay. So that will be the uh, further further development process, and it undergoes with the um, third part, that will be embryonated egg with third instar larva. Okay, and that third instar larva will ingest by means of this particular host, that is the human. Okay, and that third stage larva will be the infective stage. Okay, where at the fourth process or the fourth step is ingestion of these embryonated eggs by means of the contaminated food or the contaminated drinking water. Okay, so after that, the fifth step, okay, in the intestine. Okay, after that it get migrates to the lungs. Okay, and it is hatch larvae and circulation and migrate to lungs. Okay, so that embryonated egg is hatching into the intestine after the first contact. Okay, and that first contact will be hatching of that particular um, third instar larva egg. Okay, and from that these larvae come out from that egg and it get migrated to the lungs. Okay, the hepatic portals, and then again um, get swallowed by means of the cuffing. Okay, and again the sixth part will be showing the migrations. Okay, the seventh part will be the seventh step will be larvae are cuffed up and swallowed and re-entered the gastrointestinal tract. Okay, and maturation process in the small intestine happens. Okay, so after the seventh step, there will be, after the maturation, there will be copulation, there will be fertilization into the intestine itself. Okay, and then these are get eggs are carried out by means of the fecus. So here the life cycle is completed. Okay, so this life cycle goes continues with the, if unfertilized egg into the soil, that will not go under the further development. Okay, and if that eggs are fertilized, okay, and then they are going to get carried their path forward to the uh, fertilization, again into the embryonation. Okay, and again into the ingestion, again into the infection, again into the migration, as continues to the again that um, second generation will giving the eggs into the feces. Okay, so we are going to stop here and we conclude uh, up to the life cycle of the Ascaris lumbricoides today. Okay, thank you.